Mugal. Carina is Austrian and did her studies in Austria, but then she did her PhD and most of her previous career in Uppsala in Sweden, working on bird speciation from genetic, genomic, and also mathematical approaches. Carina has been hired at CNRS at the LBBE in Lyon. She's been there for 3 years, and she's going to be presenting today.
Yeah, so thanks for inviting me to speak here. I'm happy to present our work. So I'm interested in speciation in 2 songbirds. And so this is collaborative work, which I've been starting with researchers in Sweden, and I'm now continuing at the LBB in Lyon. And the 2 songbirds that I'm interested in are the collared and the pied flycatchers. And I'd like to start by introducing you why I think they're an interesting study system. And then I will show you 3 parts of results. One part I refer to as classical speciation genomics, which didn't show what you usually expect with classical speciation genomics. So that's why we turn to look at other approaches, which is then the second 2 parts, where we in both cases take advantage of new sequencing technologies. So why did we choose to study collared and pied flycatchers? So birds in general, they're particular research, particularly ecologists, because you can easily observe them in the field. And you can collect a lot of behavioral information and also information traits that are relevant to reproduction and reproductive isolation. So there is a long history of studying birds, and one of the systems that has been studied for decades are the collared and pied flycatchers. So we are in a position where we have a lot of ecological and behavioral information available. My collaborators, they have studied them over decades. And now what we started is to complement what we know from ecology with genomic resources. So we have a lot of traits available, and from demographic modeling, we could show that the 2 birds are actually quite closely related. They are roughly 500,000 years separated, and they are likely shaped by speciation in allopatry with periods of secondary contact, which is also reproduced by demographic modeling, where we find evidence for ancient gene flow, but no present-day gene flow. So what do we know about the birds today? So today, the birds share a large hybrid zone. So you can see the area in green of the pied flycatcher, the area in purple of the collared flycatcher, and then a huge region of secondary contact, which is in Central Europe and also on the Swedish islands of Öland and Gotland. Now, these 2 areas of secondary contact are actually hybrid zones. So we find F1 hybrids, but we don't find any back crosses or further generation hybrids. So it's now in these hybrid zones that the birds have been studied. And in particular, what is interesting to speciation research is collecting traits that are relevant to reproductive isolation. So what we know from studying birds in these hybrid zones is that we see various sources of reproductive isolation. We see pre-mating isolation in form of divergent habitat preferences, slightly divergent reproductive timing, assortative mating. We also see post-mating pre-zygotic isolation. So if you have females that have mated with both kind of males from the same species or from the other species, the egg is preferentially fertilized by the sperm from the same species. But these 2 stages are not complete, so we do observe F1 hybrids. And the F1 hybrids, they have reduced attractivity. They also have metabolic issues, so high metabolic rates. But what is quite interesting is actually that we observe complete sterility. And this is quite interesting because here's just a summary of what it just said. So we have all stages of reproductive isolation represented. The stages are not complete. And this stage, the intrinsic postzygotic isolation stage, is complete. So we see complete sterility independent of the environment. And this is interesting. Why? Because this goes against our view what we have in birds. In birds, we generally assume that pre-mating isolation is very strong, and also behavioral issues in the hybrids. So they might have low attractivity, and that this is sufficient for reproductive isolation and speciation. And what has the general picture that we see is that when we see hybrids, they actually, even after 20 millions of years, we can see that they are still fertile. So this is why we think, and we as my collaborators and me, that the flycatchers are a quite interesting study system, because they represent an exception to our general perception of speciation in birds. And then, so we have our study system identified, and this is then a classical microevolutionary approach to speciation research. So we study speciation in our study system, and we follow what basically most do in the research field. We are interested to identify the genes and genetic elements that are involved in reproductive isolation. And then, of course, we would like to reconstruct the history of the different barriers, so pre-mating, pre-zygotic, post-mating, pre-zygotic. Is it these more ecological terms that drive speciation, or is it really the intrinsic barriers that drive speciation? So I think this is the two typical modes of speciation that we have in mind, that speciation is driven either by ecological adaptation to different environments, which would then be reflected more in pre-zygotic barriers, or is it really intrinsic barriers, postzygotic barriers that drive speciation? And now we have in our study system complete postzygotic isolation, but we still don't know if these ones were the ones that actually arose first, and if these ones were the ones that drove speciation. So ideally, since we see reproductive isolation at all stages, we would like to identify all the different candidate genes and then reconstruct their history. And now we do this in our study system, and then if you want to put this in a bigger perspective, it's then also interesting to compare it to what is observed in other bird systems. So this is our study design, and we start with what I refer to as classical speciation genomics. What do I mean with classical speciation genomics? Now, here is what the general idea of classical speciation genomics. If this is on the x-axis, you see the genome represented, and on the y-axis a measure of genetic differentiation. Then the idea is that where we have virial loci, there is no gene flow between the species or incipient species, and gene flow reduces genetic differentiation. So since we have no gene flow where we have the virial loci, we have elevated genetic differentiation. So what we expect under the classical model of speciation is we expect that where we have virial loci, we have elevated FST, that's a relative measure of species differentiation. We have elevated DXY, which is an absolute measure of species differentiation, and we might have slightly reduced genetic variation within the population. So that means we can just sequence genomes of our species pair of interest and then compute the differentiation landscape, look at the differentiation peaks, and then by that identify relevant virial loci. So this is what we did. And this is a study I was involved in during my postdoc, and this is basically the results, which were a bit unexpected, made me interested to continue speciation research in this study system. But what we did back then is so this is our species pair of interest, the pied and the collared flycatcher. We sequenced 4 different populations of each of these species, and also 2 additional species, because we wanted to look at the genetic differentiation landscape along the speciation continuum. So we sampled 20 birds of each of those populations, and then we computed the differentiation landscape, which is shown here. So I'm showing it for one of the chromosomes, but it looks like that across all of the chromosomes. The interesting aspect, I think, is shown here in the center. So here we have the FST peaks for the different pairwise comparisons in the top panel. And an interesting aspect you can see is that they are actually strongly correlated among different pairwise comparisons. Also, the FST peaks between species are reflected when you look at the within species level. And then the most interesting aspect is actually the DXY. So our model tells us that where we have an FST peak, we should also find a DXY peak. And we see exactly the opposite. We see a strong valley in DXY. And this strong valley is reflected by a strong valley in pi. And that's quite interesting because that's not what you expect. So how can we explain this finding? So what we then saw is, we saw a strong reduction in pi, and that's often related to linked selection. So that would mean we should expect to find the lower recombination rate. And that's exactly what we see. Wherever we see these FST peaks, we see strong reduction or a local value in recombination, which is now shown here. So here you see pedigree-based recombination rate estimates for the colored flycatcher. And if you look at this genome-wide, we can also see that the recombination rate is negatively correlated with the differentiation landscape. So this provides evidence that recombination and linked selection shape the differentiation landscape in our study system, and that the FST peaks don't reveal what we expect. So they don't reveal the barrier loci. So why would local reduction in pi lead to FST peaks? So this is an illustration. So linked selection locally reduces pi. If you have a local reduction in pi, you have a locally reduced population size. So if you imagine you have two colors of balls and a small number of balls, like let's say only two, it's very likely that you sample only red or only white. But if you have a large population size, the likelihood like 200, it's very unlikely that you sample 200 red or 200 white. So this sorting, lineage sorting, takes much more time. The smaller the population size, the faster the lineage sorting. So this is a mechanism that has nothing to do with selection. It's just increased power of drift. So faster lineage sorting, that explains our FST peaks. So what we concluded is that in our study system, the classical model or the classical predictions that we have for identifying barrier loci are not justified. Instead, we find what we call the linked selection model. So we have FST peaks. Wherever we see FST peaks, we have DXY values, which are explained by values in pi, and the requirement is that we have a locally reduced recombination rate. Now, this is quite exciting in a way when you find something that is not expected. In addition to finding something that was not expected, but we can also conclude that our approach didn't reveal any barrier loci. So I found this quite exciting because that means, on the one hand, we want to understand why we see what we see. On the other hand, we also need to think different. What approach can we use to identify barrier loci in our model system if we cannot do what is working very well in most other study systems? And what we found is actually that it's not only not working in our study system, it's actually not working very well in birds in general. So for many birds, the linked selection model applies. So that's why I decided I wanted to investigate this further and understand better why we see what we see in our study system and what is actually explaining the differentiation landscape in our study system. So we saw from estimating recombination rate in our study system that the recombination rate is strongly correlated among the species. And now the correlation that we see between the differentiation landscape, is of course consistent with the correlation in the recombination landscape if recombination is the driving force of explaining the differentiation landscape. However, we also have to keep in mind that the black and white flycatchers, they still share many polymorphisms, so we could see a correlation also because of shared patterns of polymorphisms. So to really understand the role of recombination in our study system, we decided we want to move to more divergent species pairs and then explore the role of recombination in more detail. And this is the work of my former PhD student. We decided we compare the collared and white flycatchers to patterns observed in another pair of red-breasted and tiger flycatchers. Now, these flycatchers, they only share roughly 2% of polymorphisms. So we computed the differentiation landscape. We could see that overall, we have a much lower correlation in the differentiation landscape. And then if we look at our FST peaks, we could identify two types. FST peaks that are shared among the pairs, and FST peaks that are lineage specific. Now, if we have these two types of FST peaks, we can actually ask the question, do evolutionary changes in the recombination landscape explain lineage-specific FST peaks? Or explain evolutionary changes in the differentiation landscape? And this is the question that we wanted to address. We did this genome-wide, but I would like to illustrate it by these two types of peaks. So one group of shared FST peaks and another where we have lineage-specific FST peaks. So two chromosomes, and it looks the same genome-wide. If you now look at these two chromosomes, we see where we have the shared FST peak, we see local reduction in 5, and we see the main aspect is reduced recombination rate. Also in FST peaks that are unique to collared and white, we see local reduction in recombination rate. And now it's important to keep in mind that we have recombination rate estimates for the collared flycatcher. So this is pedigree-based recombination rate, and we only have this available for one of our species, because then you have really to get pedigree data for several hundreds of individuals. And if we summarize this genome-wide, this is now shown here. So we categorize our genome into 4 groups. So we have the FST peaks that are shared. We have the FST peaks that are unique to either of the two study systems. And then we have the remaining part of the genome where we don't find an FST peak in either of the two species pairs. And then we were interested in what's the recombination rate in these 4 classes. So of course, our prediction is we have reduced recombination rate. In the shared peaks, indeed, we find reduced recombination rate in the collared flycatcher, and also in the peaks that are unique to the collared and white flycatcher. And this makes sense. What is interesting then is that where we have FST peaks unique to the other pair, the red-breasted and the tiger flycatcher, we don't see reduced recombination rate in the collared. So this provides us with indirect evidence that evolutionary changes in the recombination landscape result in evolutionary changes in the differentiation landscape. Of course, ideally, you would have recombination rate estimates for all the 4 different species, but we simply don't have access to pedigree data for all the species. But we still wanted to investigate this further. So instead of estimating pedigree-based recombination rate, which we couldn't get for all of the species, we estimated recombination rate based on linkage disequilibrium, and then we wanted to see if we now estimate recombination. So this is basically the plot that I showed before. We're now on the x-axis, on the y-axis, you don't see pedigree-based recombination rate, but you see recombination rate in the collared based on linkage disequilibrium. So we reproduce what we saw before, now with another type of recombination rate estimate. And then the interesting aspect noise that we have for recombination rate also for the other species pairs. And then the shared peaks and the background, of course, they don't change, but these types flips. So this is a more strong evidence that really changes in the recombination rate are driving changes in the differentiation landscape. Another interesting observation we made is that we have FST peaks. Wherever we see these FST peaks, they're generally quite large regions, several megabases broad. Then we thought maybe, since we have large regions which represent FST peaks, it could simply be a reshuffling of the genome, because we only have one reference genome. We have our color flycatcher reference genome, where we map all the data. Then we thought maybe if this is a central part in the colored flycatcher, it could be moved to another part in the tiger and red-breasted flycatcher, so that this could explain why we have changes in the recombination landscape. Since we don't have a genome assembly for those species, what we did is basically we looked at the recombination landscape based on the distance from the end to the center. And then we saw that it's not reshufflings. There is no big rearrangements that explain, because the recombination rate averages are basically reflected. In birds, you have two classes of chromosomes. You have what is referred to as macrochromosomes, and then you have smaller chromosomes. So we had to separate them in two groups, because the trend of recombination rate is different for the two groups of chromosomes. But we can see it's not a reshuffling of regions. So something that is internally in the colored is not that all of a sudden at the end in the tiger or in the red-breasted flycatcher. This was suggesting it's not rearrangements, but we still thought if it's these large regions, it could be structure variants like inversions that are segregating in the population that could locally suppress recombination rate. So this is now our main conclusion from this work, was that, okay, in our study system and in birds in general, the differentiation landscape is shaped by linked selection, and evolutionary changes in the differentiation landscapes are not explained by lineage-specific adaptation like in most other study systems, but they are explained by evolutionary changes in the recombination landscape. And our idea was that it's broad scale, since we see this broad scale local suppression of recombination rate, that this could be caused by segregating structural variants like inversion polymorphisms, which are suggested to play a role in locally suppressing recombination rate. And then another conclusion from this work is that we could identify a role of genomic architecture in shaping the differentiation landscape, but we couldn't use this approach to address our initial question. That's identifying the barrier loci and study the evolutionary history of speciation. So we basically ended up with two questions. On the one hand, we wanted to understand what we see better. So we wanted to see what molecular mechanisms underlies the local suppression of recombination rate, and which approach can we use in our study system to identify barrier loci. So to address the first question, since we had our idea that structural variants play a role, we said we want to look beyond single nucleotide variants and really assess structure variation in our study system with the help of long-read sequencing technologies. And then to identify barrier loci, we decided we wanted to integrate functional genomics into our research. So let's first look at our results from the first question. So our main hypothesis is, as I said, local suppression of recombination rate is a result of structure variation, like, for example, inversion polymorphism segregating within the population. So to test this, we decided to do long-read genome sequencing and to assemble both species. So first we had only one species assembled, the colored flycatcher. Now we generated genome assemblies for both species. So this we did with a combination of PacBio long-read data and high-C, so three-dimensional data that are helpful for scaffolding. And then we also generated population level long-read sequencing data for 10 individuals each. Now the genome assemblies, and that's again the work of my former PhD students, are illustrated here. So we could actually, what this basically illustrates is these blue squares illustrate the scaffolds, and what we could see here is that the scaffolds really represent basically chromosomes. So we could generate a chromosome level genome assembly based on this data. This is, for example, scaffold N50 basically represents the same number as chromosome N50. And then we are interested to identify structural variants. So how did we do that? We used two approaches. On the one hand, we identified structural variants simply by aligning our two genome assemblies to each other. We identified roughly 40,000 structural variants, which span 60 megabases. That represents 5% of our genome. And then we grouped the structural variants into insertions, deletions, duplications, contractions, translocations, and inversions. So you can see that translocations and inversions, they are much rarer, but they tend also to be much larger on average. Now, these structural variants are distributed across autosomes, the large autosomes and smaller autosomes, but they're also found on the sex chromosome. Two interesting observations that we made is that we have a higher density of structural variants on the smaller chromosomes than on the larger chromosomes. And when we look at sex chromosomes, we see that we have an enrichment of inversions on the sex chromosomes as compared to the autosomes. Now, our main question was, is there an association of FST peaks and structural variants? And our prediction was that we find a positive association. That would mean an odds ratio significantly larger than 1. And this is not what we observe. However, if we split up our structural variants into the different types, insertions, deletions, duplications, contractions, translocations, and inversions, then we actually can make an interesting observation, which is quite well in agreement with our hypothesis. We see a positive association of translocations and inversions with our FST peaks. And it's actually translocations and inversions that are suggested to play a role in local suppression of recombination rate, and not so much insertions and deletions. So what we observed was actually making also sense from expectations. But so far, these structural variant calls are based on comparing two genomes that we have assembled. So we actually don't know, do the structural variants represent fixed differences between the two species, or is it segregating polymorphisms? Now, to differentiate fixed differences from segregating polymorphisms, we used our population level long-read sequencing data, and our idea is that it must be segregating polymorphisms, because what is a fixed difference is no longer going to suppress recombination rate in our study system. It's only when a structural variant is in a heterozygote state, it's going to suppress recombination rate. So it needs to be a segregating polymorphism. And this is now shown here. So here is still our values from the SV calls from comparing the two genomes. And now we have added the structural variant calls from the population level data. And what you can see here is, we see the same trend. No, so that's what we would expect. Translocations and inversions are much rarer than insertions, deletions. Then since different structural variant callers differentiate in different ways between inserts and duplications, contractions, we decided to merge this category. And then from this category, we were then interested to identify which ones are fixed differences between the two species. Here we identified roughly 550 insertions, deletions that were fixed between the two species. And then when we merge these together, I merge these together simply because the ones that we identify are so rare. And then it depends what kind of structural variant caller you use, which one you identify as fixed. So that's not always the same. So I give you a number which is smaller than 5, it can be 1. If you use one structure variant polar, it can be 4 if you use another one. So I decided I will not give you a precise number. But it's smaller than 5, but that's still helpful for addressing our questions. Then we were interested, is it these ones or is it the other ones, the ones that are still segregating as polymorphisms that are associated with our FST peaks? And we can see where we find actually a fixed inversion or translocation, that's not where we find an FST peak. So it's really where we find segregating polymorphisms where we see the FST peaks. So we find an association, as we hypothesized, between FST peaks and segregating inversions and translocations. Now, of course, it would be nice to also get estimates of recombination rate, more direct estimates of recombination rate for both species, not only the collared flycatcher, but also the pied flycatcher, to really investigate the association of segregating inversions and translocations more directly. And that's a work in progress. So we are currently generating direct recombination rate estimates for both of these species. And then we want to see if local suppression of recombination rate is happening in those individuals where we see heterozygote structure variants. So this is still work in progress. And then another aspect that we saw in our study system is that they're actually quite closely related. So we could generate a list of fixed differences between the two species, single nucleotide variants that are fixed different between the species, but now also insertions, deletions, inversions, and translocations. So this provides us an interesting candidate set to investigate if these fixed differences play a role in our speciation processes. So we are currently annotating the assemblies and our fixed differences and exploring if these fixed differences play a role in the speciation process. This takes us then to the next question, where we then integrate functional genomics into our research. What do I mean with functional genomics? So we conduct a functional annotation of our fixed differences and see if we can identify some interesting genetic elements that could play a role in the speciation process in our study system. And we also integrate different types of omics data to investigate the role of gene regulatory evolution. So if you look at the first part, so as I said before, our two species, they're quite closely related to each other. They're roughly 500,000 years separate. Now, if you have closely related species, this should be reflected in a low number of fixed differences also at the molecular level, and that's what we see. So I'm not just starting by looking at the fixed differences at the single nucleotide level. So we are only looking at fixed single nucleotide variants. We annotated them. And then we found we only see fixed differences in 303 protein coding genes that have an influence on the protein sequence. So this is actually a quite small data set that you can start to explore. And that's what we did. We wanted to see, do we find something particular among those genes? So we do classical gene ontology analysis, gene ontology enrichment. And this is what we saw. So most of these terms are actually related to meiotic cell division. And that's a very strong overrepresentation. And that's, of course, quite interesting because we have a study system of birds where we see complete intrinsic postzygotic isolation in form of sterility. So something could go wrong simply during gametogenesis, during meiosis, which could explain the sterility in our study system. And that's what we wanted to explore in more detail. So the hypothesis is that differences in interacting protein coding genes during meiosis could lead to patient-Dobzhansky-Miller incompatibilities in our F1 hybrids that could induce meiotic arrest. So to address this in more detail, And that's now not a study that is led by me, but it's led by my collaborator, Anna Kornström, an ecologist at Uppsala University in Sweden, and her former PhD student, Carina Mugal. So what Carol did is she had single nucleotide RNA data from the testes, and then she used the expression pattern of the single cell data to cluster them based on their types. So based on their expression patterns, and you see different kinds of clusters, and then based on the genes that are expressed in these clusters, we can identify to what stages of spermatogenesis these clusters correspond to. So we only had the samples from males. That's why it's spermatogenesis. So we were able to say which clusters correspond to which stages. And now we wanted to see in what stages are the genes that we have identified to have fixed differences in their protein coding genes expressed. And this is now shown here. So we take the genes that are expressed during the different stages, and now the stages are ordered based on spermatogenesis. And you can see, in particular in the earlier stages of spermatogenesis, these genes are expressed. So this could mean that it is consistent with the idea that Bayesian-Dobzhansky-Miller incompatibilities in these earlier stages could lead to meiotic arrest. This is still ongoing work, but what we then also saw, what Caro found in her data, now she added data, single cell RNA data for hybrids, and then she wanted to see which cell types are underrepresented. And it's exactly those later stages that are underrepresented in the hybrids. So that's also consistent with the idea that something goes wrong in the stages where our proteins of interest are expressed. And this is also confirmed based on histology, where we also see that the later stages of spermatogenesis are absent. But this is still a work in progress, but all looks very consistent with the idea that these genes could play a role in explaining the sterility in our study system. And then we also wanted to see, because this not only addresses the sterility in our hybrids, but we also wanted to see, can we use other approaches to identify genes of interest? So we integrate gene expression data and investigate the role of gene regulatory evolution during speciation. So first we started by looking at the expression divergence between species, but we also look at expression patterns in the hybrids. And why is it interesting to look at expression patterns in the hybrids? So often we might expect that expression is dysregulated or transgressive in the hybrids. That means the hybrids lies outside the range of the parental species. So it's either higher than both of the parental species or lower than both of the parental species. And why is this something of interest? Now, gene expression is regulated by the interaction of cis and trans regulatory elements. So we have again several elements interacting with each other. So that's a good candidate for Bayesian-Dobzhansky-Miller incompatibilities, these regulatory elements. That's why expression patterns in hybrids are actually quite relevant to speciation research. So the idea is that when you have divergence in cis and trans regulatory elements between the two species, of course, if you have a cross, one trans regulatory element can find its corresponding cis regulatory element and everything would be good. But this is not always happening because also the other pairing can happen, no? And then depending if this is now an enhancer or a silencer, the expression in the hybrid can be lower than the expression in the parent or higher in the hybrid than in the parents. And any of these combinations can happen. So this will then lead to transgressive expression patterns in the hybrids. So to investigate this, we are restricted to studying natural hybrids. So we have hybrids that we can find it's great if you can study natural hybrids. That one aspect when you study natural species that you don't have control of everything. So you often don't get the trials. You don't get adult hybrids and where you know the parents. So if we sample adult hybrids, which we wanted to sample because we wanted to have hybrids that have testes, then we didn't know the parents of those hybrids, but we decided we sampled males, adult males from pipe and from collared, and unrelated hybrids. So we have this kind of study design. Then based on mitochondrial DNA, we could see that we also always have one crossing when we sample adult hybrids in our study system. It's always one direction of the cross. The pied is the mother and the collared is the father. So we always get this kind of cross, and we sampled randomly. So this is just when you study natural species, you have not so much influence than when you study lab species. And then we generated transcription sequencing data for 5 tissues. These 5 tissues are brain, heart, kidney, liver, and testes. So the testes is interesting because we have sterility phenotype in our hybrids. But first of all, we just started by looking at differential gene expression between the 2 species. And then you can see something which I think is more or less what you would expect, that the brain, and this is now a particular region of the brain because we have quite some heterogeneity in brain region. So we focused on a region which we think is actually relevant for song learning, and that's quite interesting in birds. So we restricted it to this region, and this is most conserved, and this is what you would expect, that brain expression patterns are more conserved. Tissues like heart, kidney, liver fall somewhere in between, and the testes is the most divergent. And this gives us a candidate list of genes of interest. My collaborators are investigating their role in the speciation process. What I was more interested in this study is also to see what explains the regulation of gene expression evolution. One observation that was interesting is that the differential expression is actually tissue specific. We only have one gene that is differentially expressed in all of the tissues, and that's a gene that is partially deleted in the pied flycatcher, and that's why it's always not repressed in the pied flycatcher. Then you can use allele-specific expression in the hybrids to see if it's cis or trans regulatory divergence that explains the divergence in the gene expression. Because if it is cis, then you know that the allele that is higher in species A, allele A, should also be higher expressed in the hybrid. Because there is a direct association with the cis regulatory elements to each gene. So you can see if the differential expression patterns A higher than B or A lower than B is reflected in the allele-specific expression in the hybrid. So if we do that, we saw that we saw an association with that. And now I would say this, we only see it in kidney, liver, and testes. I think this is a power issue because we have very few differential expression in the other 2 tissues. So we see an evidence that cis regulatory divergence plays a role in explaining the expression divergence in our study system. We also investigated it based on another way to see if we can find fixed differences in proximal conserved non-coding elements. So that's regions that are in the cis of the or upstream of protein coding genes, which are strongly conserved, which could play a role in gene regulation. And then we only see an association for testes, but I would again say this could again be a matter of power because we have the highest power in testes where we find the strongest or most differential gene expressed. So we find evidence that cis regulatory divergence plays a role in the gene expression divergence that we find in our study system. So this is now when we focus only on the comparison between collared and pied flycatcher. But as I said, we also wanted to look into expression patterns in hybrids. So what did we find here? So we were interested to identify transgressive expression patterns. So gene expression that is higher or lower than both of the parental species. Now what you see here is that there is so now you see comparison hybrid versus colon, hybrid versus pied. Now if you see differential expression, that doesn't necessarily mean it's transgressive, because if the parents are very divergent and the hybrid falls in between, you will get significant differential expression, also if something is in between. So in the testes, all of the 61 genes were in between. There was zero evidence for transgressive expression in our hybrids, but we found quite some evidence for transgressive expression in heart, kidney, and liver. So most of the genes were actually transgressively expressed. That's quite interesting. What we had expected, since we have the sterility phenotype, is actually that we would find a lot of transgressive expression patterns in the testes, and that's also what is observed in many other study systems, that sterility phenotypes are related to transgressive expression patterns. Now, here it's important to keep in mind that the testis is actually a compound of different cell types. So it could actually be that we see transgressive expression patterns, but that the different cell composition would mask this. And this is, since we have the single cell data from the other study, this is actually what we can confirm. This is still ongoing work, that we see a lot of differential transgressive expression in different cell types, in particular in those cell types that we think could play a role in the sterility phenotype. But we don't see anything if we use bulk data. So that was quite interesting also from a methodological point of view. So we looked at the expression patterns, and I think in our study system, this was much more promising to identify loci that are interested in explaining the evolutionary history of speciation than using a classical speciation genomics approach. And then finally, we also wanted to understand the role of genetic versus epigenetic changes in regulating the gene expression patterns during speciation. So that's what we did. To the same samples that we had sampled before for RNA-seq data, we added whole genome bisulfite sequencing data. That's another student that worked on that. And then we wanted to see what's the role of methylation in gene expression regulation in general. So for this, first of all, we just classified the genes into low, medium, and high expressed genes and look at their methylation level. And then we can see some patterns that, in particular in the promoter regions, and this is maybe what's expected, we see if we have CpG alleles in the promoters, we see low expression levels. So we split our genes in two categories, those with CpG alleles in the promoters and those not. And then you can see that the reduction in methylation level is generally observed where we have CpG alleles, and this is negatively correlated with expression level. So since you don't see this very well like that, we also just computed the correlation along the gene. Now you see the correlation between methylation level along the gene body for the different tissues and the different species, but there is no difference among species. So it's mainly the concurrent that are indicating different tissues. What we can see here is that in genes that have a CpG allele in the promoters, you see a strong negative correlation between methylation level and expression level. So that means if methylation in the promoters plays a role in expression regulation, then it could play a role in expression divergence. And that's what we wanted to investigate. So we did that, again, with using profiles. So we computed now not differential expression, but differentially methylated region. It's the same principle. So this is now shown on the y-axis, and we computed a profile along the different genes. And then we separated our genes into two categories, those that are differentially expressed and those that are not differentially expressed. And here we can see that those that are not differentially expressed generally have it's significantly lower differential methylation between the species, which is basically that the dashed lines are significantly below the solid lines in the promoter region. This is for epigenetics. Then we wanted to see how does it look at the genetic level. So we already before saw this association, the differential expression was associated with cis regulatory variation. Now we just did it again along the profile to see what we see if we use such an approach. So now the non-differentiated genes, differentially expressed genes, are significantly below the differentially expressed genes for all tissues. And it's actually not only upstream, we see it in the entire region. Was an interesting observation, but it's of course in agreement with what we saw before, where we mainly focused on the upstream region. So we can see an association between methylation divergence and genetic divergence in gene expression divergence. Now, how does it look with the transgressive expression patterns in our hybrids? Are they also explained by epigenetic patterns or genetic patterns? That's what we wanted to see. And this is now shown here. And here you can see that the solid and the dashed lines, they fall all over the place, and there was actually no statistical association. So while we see an association of genetic and epigenetic patterns with gene expression divergence between species, transgressive expression patterns seem to be regulated by another mechanism. And with that, I'd like to conclude the final part. So what I think was quite exciting is that when we integrate functional genomics approaches, we actually find some interesting patterns that could explain the history, the evolutionary history of speciation in our study system. So if we focus on the fixed differences, and what I showed you so far is only the fixed differences that we found in protein coding genes, we could actually identify interesting candidates that could play a role in the breakdown of meiosis in our study system. And the integration of transcriptomics data in general can be useful to identify different types of genes that could play a role in the speciation process. So that's the patterns that we observed across the different tissues. We also find different expression, for example, in the brain. And if we follow this up, which one of my collaborators is doing, we are investigating if these actually play a role in the song learning or song divergence in our study system. So if we wrap up all the three different parts, then I think one of our main conclusions from the first part is that the classical speciation genomics approach didn't give us the results that we were expecting initially. We saw a strong role of genomic architecture in shaping the differentiation landscape, but this approach didn't reveal us any interesting barrier loci. I think that was actually quite exciting because that pushed us to use some other approaches. So we integrated functional genomics, but it's also interesting to explore in more detail what's responsible for the local repression of recombination in our study system. And with that, I'd like to thank all the people that contributed. So it's former students that work with me in Uppsala University in Sweden, and some colleagues also in the YOM who got involved, helping me out with the analysis, and all the people that have been contributing to field work and also some molecular work, funding, and thank you for your attention.
We have time for questions.
Very nice, very interesting. Looking into the detail from the molecular. I'm interested if you have a hypothesis of why you don't observe any irrigating since that, since you said that this is kind of what you expected according to the classical studies?
So we do observe pre-mating isolation in our study system. So we have divergent habitat preferences, we have associative mating, but it's not complete. So we have roughly 6% interspecies mating. It's expected that this is strong, but our study system is actually so young. So often when we compare bird species and we say, well, after 20 millions of years, we still find fertile birds, we are on a different time scale. We are looking at much more divergent birds. And then pre-mating isolation is strong enough. Maybe, I mean, now I can only speculate because you can test this in natural systems. Maybe if we wouldn't have postzygotic isolation that has evolved so quickly, the pre-mating isolation would evolve to be more strong in the future. But we cannot test this any longer. But it's not against the pattern that is observed in the other species. The only thing that is unusual for birds is that postzygotic isolation evolved so quickly. Or that's at least the general perception that we have. It could also be that our general perception is wrong, because maybe it's very difficult to identify natural species that hybrids are actually sterile. Because yeah, they could also be inviable, then you don't find them. And then you find some that are viable, that then also are fertile. So we could have a biased sampling because we studied natural species.
Thanks for talking about the first part. Was close with the expectation with respect to whether the, let's say, the inversion that you find polymorphic or fixed. Because if I understand correctly, you're suggesting that it's a complete linear sorting that is different with respect to whether the relation is higher or lower. That explains the right? That's what you
Fast speed up in lineage sorting, yes.
faster lineage sorting.
Yeah.
Okay. But with respect to current recombination rate, not so sure it matters if you imagine an inversion that fixed in the past and is currently fixed, and so because it's fixed, recombination rate is not altered. Yet during the process of fixation, you had an episode where you actually fix a piece of a chromosome, which is an episode of very strong drift and linear sorting. And so it's a bit bizarre that you don't find any
So
strong signal for the fixed one.
Yes, so of course, the ones that are fixed now, they were polymorphic in the past, so they could have played a role. Then I think, and we need to investigate this in more detail, that it's the allele frequency that matters, because it needs to be an allele that is maybe intermediate in frequency, because if it's very frequent or very rare, it occurs very rarely in a heterozygote state, and then the local suppression of recombination rate might not be very relevant. So that's work in progress, that we need to investigate the allele frequency, the role of the allele frequency in the segregating polymorphisms. Then the ones that are fixed are very few, and maybe that's the ones that are already very differentiated at the relevant time point. So, but we need to investigate this further, yes.
Thank you for the presentation. I have a technical question because I'm not familiar. How do you do from pedigree to estimate recombination?
Well, you sequence, what we had, we did genotyping. So we have markers spread along the chromosome. And then basically you look at inheritance, and it's a statistical approach. So you know we have several generations in the pedigree, and then you can track markers that are shared together all the time. Those should be genetically linked. That's basically the method you use to see what kind of markers are statistically significantly shared.
And you suggest this is the most the best way, the more robust?
No, I don't say it's the best way. It's one way that is good for population genetics analysis. I prefer it in comparison to linkage disequilibrium because it uses an independent data set. So if you have a, if you, for example, identify sweeps or FST peaks from SNP data, and then you use the same SNP data to estimate recombination rate, that could be confounded in some way. We still use it, no, because we didn't have other estimates available, but then it's not independent. So I only think it was nice to have pedigree data because they are independently generated. But the other ones are there, and we tested, and we found the same pattern. But if you only have this kind of data, you will be questioned quite a lot if you try to publish it without being able to validate that this, because they are dependent data.
I also have one more basic question. How do you know it's a hybrid?
Well, so I know it from the genome because
but-
the biologists know it also from the singing pattern. For me, this is so you can.
Yeah, because my question is like, why maybe it was not a hybrid?
Yes. So you go into the field. This is how we did it. You go into the field. You identify male hybrids based on their singing pattern. And that's when you're a good ecologist, you can do that. I cannot do it, for example. I'm not a good ecologist. I focus more on the genome data. So then I take the genome data and I have identified a list of fixed differences. So I trust my collaborators that they give me hybrids, and then I double check it based on the genome. I have a list of fixed differences at the single nucleotide level. So those that are fixed different between the two species, they need to be heterozygote in the hybrids, and I saw that.
And there's no error? What's the error rate?
No,
Really? They get it so...
They get it very well. So it's very impressive. Yeah.
Try to do kind of a random check, like take randomly also things that are, they would say it's not a hybrid, to see if it's really, always...
So, I mean, I think if you've studied the birds for a long time, you learn how to identify them. Then also often the phenotype is a bit intermediate as well, that's confirmatory, but the intermediate phenotype can actually also happen even if it's not a hybrid. So the song pattern is quite strong. So, I mean, when I listen to the recordings, and then I can also I can also see it. But in the field, I find it more difficult because you have all these different other noise. But if you have done it many times, so they identify the hybrids, and I check it genetically, and they were all hybrids. They were all heterozygote. And then we use actually the mitochondria, because the mitochondria is maternally inherited to identify the direction of the cross. So that's so then we know.
I have another one. In the second part, you didn't talk much about the female F1 hybrids, and I was wondering whether you had evidence for meiotic disruption females or.
So we know that the females are also fully sterile, and then based on Haldane's rule, we would expect that the female is actually sterile before the male in birth because it's the heterochromatic sex, and we see it. Then to sample, so there's two issues with studying females. On the one hand, identifying the female hybrids is more challenging in the field because they don't have the song patterns. Then we want to sample hybrids, but then we want to sample hybrids that if we want to sample female hybrids, we can cross them in cages, but then we have young ones. So the birds, they develop the full reproductive organs in the reproductive season. So need to be the adults. Then with females, you also have another challenge that spermatogenesis is happening all the time. And in females, not. So to get sufficient meiotic cells in the females, so you have several challenges with the females. We want to try, but we started with what was easy. So that's why we started with the males. But we definitely want to try to isolate also cells for females.
There's no questions on that.
Okay, so one more questions. Thank you, Carina, for the So